This is Sandra Porter from Digital World Biology. Today, I'm going to demonstrate how we can use Molecule World to find interactions between amino acids in the same protein chain. Interactions between amino acids contribute to protein folding, help maintain a protein structure, and control a protein's activity. Today, we're going to look at amino acid interactions in p53. The p53 protein is a transcription factor that forms a homotetramer and binds to DNA, as you can see in this image. Binding to DNA by p53 can induce gene expression. The p53 protein is also known as a tumor suppressor because it turns on expression of proteins that can stop the cell cycle or trigger cell death. This activity prevents tumors because it puts a check on uncontrolled cell growth. Some of the mutations in the p53 protein can lead to inherited cancers because they interfere with the activity of this protein. We're going to investigate how a mutation can affect one of the amino acids in this protein and lead to a form of inherited cancer by looking at the normal role of this protein in the p53 structure and see if we can figure out what it does. The amino acid we're going to investigate is a leucine at position 265. First, we'll find this leucine in the protein structure. We begin by opening the sequence viewer. Then we'll scroll through the sequence and look at amino acids every now and then to figure out where we are. Eventually, we will find leucine 265. Next, I touch the Show Hide menu and choose Show All Atoms. This helps me see where the leucine is located in the protein structure. Now, I'd like to see if that leucine could be involved in an interaction between the different subunits. To test this, I'm going to invert the selection. Inverting the selection deselects the leucine and allows me to change the drawing or coloring style of all the other components of the structure. I choose molecule so I can see the different subunits of the protein and see where the leucine is located relative to other subunits. Now I'm going to zoom in and see if I can look more closely at the leucine and figure out which amino acids it interacts with. I changed the drawing style to residue, and I invert the selection again to highlight the leucine, and I changed the drawing style to space fill, making it more easy to see. Now I zoom in and turn the structure to see which part of the protein is closest to the side chain for my amino acid. To identify the nearby residues, I invert my selection again so that I can see the colors better, and I use the color key to figure out which residue is nearest. From the color key and the position in the structure, it looks like a glutamic acid or aspartic acid, perhaps, might be closest to my leucine 265. I'm going to see now if I can find the glutamic acid that's near the side chain for the leucine. I apply the rainbow coloring style to help me do this. The rainbow coloring style colors proteins or nucleic acids from the 5' or amino end to the 3' or carboxyl end. It starts with red and ends with violet, like the rainbow. Now that I've found the glutamic acid, I show the side chains and change the drawing style to spacefill and coloring style to element. I can see that carbons in the glutamic acid backbone could be forming a hydrophobic interaction with the carbons in the leucine side chain. This interaction looks like it serves to hold the folded chain together. Mutations that replace this leucine with less hydrophobic amino acids or amino acids with shorter side chains could disrupt protein folding in this region and impair protein activity.